Jim McNeil, I'm the chair of the committee. The person who's the chair of the committee is the one who knows the least about the subject matter. So that's why the chair is me. We're here for the PhD Thesis Defense. The title's up there. I'd like to have the committee members just introduce themselves to the audience.
Matthew Posowitz, John's principal advisor over the last 4, 5, 6, 7 years.
Okay.
John's co-advisor for the last 11 years.
Ron Cohen, just a humble member of the committee.
Marie Gerardi, the person responsible for reading John Colorado.
Okay. And Michael in the back.
Yeah,
You are?
personally can't seem to find a legal parking place.
Yeah. They don't give parking permits, they give hunting permits. Okay, I think the way this will work is John will give a 45-minute presentation. We'll accept questions of a simple, short sentence clarification nature, but any substantive question, please wait till the end.
Okay, thank you all for coming to my defense. Hopefully you don't have too long defense. The presentation outline, basically I'm going to go over just the current state of biofuels in the United States and outlook for the future. Background on algal hydrogen metabolism and the tools that we employ to measure hydrogen metabolism. And then the specific work that I've done, first looking at diversity in various species, specifically 6 different strains of eukaryotic algae. And then the F-cluster hydrogenase of chlorella variabilis and C64A. And lastly, Cyanophora paradoxa as another algal species, which is very interesting hydrogenase. Then I'll go into the work I've done disrupting hydrogenases in Hypomonas reinhardtii. And finally, a little bit on the Clark electrode that we've developed in the lab and acknowledge any questions. So first, just an overview to get us all on the same page of biofuels in the United States. But first came here, John Trepney was the president, and he had this in the Denver Post. I thought it was very apt. It's an outmoded notion that reliable and affordable energy, a robust and growing economy, and a clean, well-protected environment are mutually exclusive. That's a beautiful statement, but how are we going to actually get there? And so this is how the DOE classifies the 3 major groups of biofuels. They've done it based on a timeline that they predict, and first generation, second generation, and third generation biofuels. In reality, it may be a little more complicated than as simple as clean as first, second, and third, one after the other. Principally, first generation biofuels involve vascular plants, seeds, and fruit. This is what many of us are familiar with in terms of the ethanol that's available at the pump today. Biodiesel, it's made from vegetable oil. I've had my own forays in biodiesel, as many of you know, with this small trailer-mounted trans-esterification reactor built with some students here at Mines. I used to power my Volkswagen Jetta. But one of the major questions that comes into play is how much energy goes into producing the fuel and how much fuel energy comes out of the fuel that comes out of the pump. And what this shows here is the energy value on BTUs per gallon. And if you don't know, a gallon of gasoline has about 110,000 BTUs. And obviously there's some conflicting data here. But overall, there seems to be a general consensus that around 20,000 to 40,000 of BTUs can be recovered from that. So 60 to 80% of the energy that is in the final gallon that's pumped at the gas station went into it in the first place to make the fertilizer, to run the center pivots, irrigate the land, and so forth. Moreover, how much actually can we produce with higher plants? This is something that I've been interested in because it brings to the point of how far can we go with higher plant biofuels. And if you look here on the left, this is basically the current vegetable oils, animal fats, and total oils produced in the U.S., which is close to 5 billion gallons per year. And if you look at just diesel alone, all total diesel sales in the United States, sorry, this is from 1999, but currently it's around 60 billion gallons per year. And gasoline alone is another 120 billion gallons. So even if you took every single acre of farmland in the United States and grew nothing but canola or nothing but soy, at the maximum efficiency you can produce oil from these crops, you'd still barely get to 50% of even just the diesel demand, and maybe a quarter of the total demand for fuel in general in the United States. So obviously, first generation biofuels aren't just that. They're a first step. It's interesting. but they're not going to do it for us in terms of our total demand, unless we make huge sacrifices. The other option is second generation biofuels, which is lignocellulosic feedstocks, and these include crop residues like the 8-foot stalks left over after you harvest the corn, or feedstocks that are grown particularly for their cellulose, like miscanthus or poplar. Basically, this is partially limited by the byproduct of other things that are in the stalks of these higher plants, like lignin, which make it difficult to break down these long chains of sugars that make up the hard cellulose, commonly seen paper. But the other limiting factor is the cost of these cellulase enzymes and the other processing. And while these plants do exist in the United States, as far as I know, there's none that are commercially viable at this time. So that brings us to third generation biofuels, which is the focus of this talk and my work here at Colorado School of Mines and Enron. And third generation biofuels relies on microbial phototrophs to provide a higher efficiency in the products that we want. They're primarily composed of lipids, carbohydrates, proteins, and various ash and other things that are these microbes. And under certain conditions, they can be induced to, in this case, be almost completely lipid that's tagged triglyceride and starch. So there's been a huge amount of interest, especially in the last 10 years, of revisiting this technology. And what's also interesting about this third generation biofuels is the various different kinds of products that can be produced from microbial phototrophs. Those include carbohydrates, lipid, protein, and then direct fuels coming directly excreted from the algae or cyanobacteria. You can make then alcohols, straight vegetable oil, and triglycerides, biodiesel and green diesel, potentially methane from the protein, and again, these directly produced fuels. So what does it all come down to, though, is how much land is it really going to take to do this? And that's where microbial phototrophs really shine, is the potential in terms of the space it would take to meet our fuel demands. So this is a revisiting of an earlier figure that I'll show in a moment. But what it shows here is basically the 2006 corn crop. And the amount of corn that you would have to grow if you were taking the grain and making it into ethanol for our current U.S. gasoline demand. Obviously, this isn't going to be possible. Cellulosic ethanol, so this considers switchgrass produced, grown on land and used as a feedstock for producing sugars from the cellulose. And again, it's pretty large, maybe the size of New Mexico. Similarly, in this case one, which is a 30% efficiency of converting the byproducts of microbial phototrophs into usable fuel, it's about the size of New Mexico. In this case two is a 70% conversion efficiency of the photosynthetically generated byproducts. So in both cases, even under microbial phototrophs, which are considered the best option, it's a lot of land if we're going to continue with the demand that we currently have. So these can be broken down kind of into first, second, and third generations. So why hydrogen from algae? Why would we be interested in that? Well, it mainly comes down to the solar conversion efficiency possible, producing a fuel directly from photosynthesis rather than having to convert it downstream. And if you look at the production from higher plants, sugarcane, for instance, is 0.16%, or if you're making a direct carbon-based fuel, so if you're using the byproduct made from plants directly, the C3 plants, or in the case of C4, where they actually have carbon-concentrated mechanisms, you might get to 6.7%. That's just producing the carbon directly, or fixing the carbon directly. If you actually have to convert that carbon then into a fuel, you're going to lose at least 50%. In the case of the previous figure, they have case 1 and case 2, where they say 30% to 70% is kind of high and low end. So then you're going to get down to this range of, maybe people have said that maybe a 1% conversion efficiency for sugarcane. In the case of producing hydrogen directly from algae, directly from photosynthesis, there's a potential to get to 10 to 13% conversion efficiency if some of the challenges can be overcome, which allows for a much smaller land area. So this is a figure from 2008, which was a chapter written by some of the authors in this room. And it shows very similar information in terms of corn ethanol, cellulosic ethanol, number 3 from switchgrass, and then number 1, which is the 10% efficiency hydrogen photo production. And even with some of the other figures that people have thrown around, like a 3.3% efficiency, you'd still only have maybe a 3 times that. So this is about 100 miles by 100 miles, number 1. If you compare that to the previous graph, which it was about the size of New Mexico or so, maybe a little bleeding into Colorado. This is a much more tractable amount of land to produce that amount of fuel. And you also have to recall that this is only for gasoline. If you include diesel, it's about 1.5 times larger as well. So obviously conservation is going to have to be the #1 imperative, even in the best case scenario. But the reason we're interested in hydrogen bioproduction is because of the potential for a much smaller land use. When it comes down to cost, there's been a few analyses done on hydrogen photoproduction. And there was an earlier analysis, this Amos 2004, which had a very wide spread between high and low values depending on the constraints that were used for the analysis. And then later, there was another analysis done, it was funded by NREL, where they really took into account many more of those constraints and defined many of those variables better. We see a smaller range of 299 to 815 per kilogram. Well, you might be asking yourself, well, how much is a kilogram in terms of a gallon? How many kilograms do I need to power my car? And so I looked into the energy density of hydrogen. And actually, hydrogen has a very high energy density per kilogram. So this is megajoules per kilogram energy density. And so it's about 3-fold higher than these other typically used fuels. So if you look at these prices here, 13 or 815, if you consider that kilogram of energy is 3-fold higher in terms of amount of energy available. And then if you're using it in a fuel cell, which has 80% conversion efficiency relative to the Carnot efficiency of the car, which might be 23 to 30%. These are, just the fact that these numbers are in the realm of possibilities is a reason to move forward. However, it's also important to point out this here is very visible, 0.01, so that's the actual energy per liter. And this is where one of the troublesome aspects of hydrogen is that the actual energy per liter of volume before it's compressed is about 1,000-fold lower. So you take 3,420 liters of hydrogen to equal the energy of a liter of gasoline in liquid form. So of course there's going to be issues with the hydrogen infrastructure, like pressurizing it, dealing with an explosive gas. And a lot of these considerations will have to be overcome. They're very important considerations in considering the hydrogen economy, even if you could produce that amount of energy efficiently. All right, so I'm ready to move on for all. Understand the reason why hydrogen might be of interest to us from phototrophs and move on to the work. So the primary research emphasis has been on the problems that have come from producing hydrogen. It'd be great to produce hydrogen efficiently from microbial phototrophs. The primary issue is that the hydrogenase itself is oxygen sensitive. And because photosynthesis produces oxygen, we obviously have a problem having sustained biohydrogen production from life. So basically algae will produce hydrogen for a few seconds to a few minutes after you've taken away the oxygen and go into an anaerobic fermentation. And this has been noted since the 1940s. But only recently, in 2000, it was found that you could deprive the algae of sulfur, and under those conditions, the algae basically has a much decreased level of photosynthesis to the point where its photosynthetic activity is below the level of respiration. So the oxygen it's consuming as it's consuming some of the sugars it's producing is actually more than the oxygen it's producing from photosynthesis, transitions to an anoxic state even in the light while it's doing photosynthesis, and it can actually sustain the production of hydrogen for a few days. And this was the first report of sustained hydrogen photoproduction and garnered a lot of attention. However, because photosynthesis is limited under these conditions, it's hard to get the levels of photosynthetic conversion efficiency we'd like to get to make this economically viable. So specifically what I've worked on here is to survey other species than the model species that have primarily been worked on, which is Ionomonas reinhardtii, and then examine another species which recently had its genome published, and specifically one that has a very unique hydrogenase enzyme. And then the last project has been to disrupt both Ionomonas hydrogenases to determine the independent roles and then provide a genetic platform for later transgenic expression of other hydrogenases which might be able to be run during photosynthesis. So I'll give you a little more background on the types of assays that I've done to actually resolve these different projects. And so basically you can break down these kind of assays into in vivo or in vitro hydrogenase activities. So in vivo activity is the activity of the cell is actually able to do. And that includes hydrogen production fermentatively in the dark, the same way yeast might make ethanol, or hydrogen production in light, which typically is transient, but under sulfur deprivation can be maintained for a few days. The other case is in vitro hydrogenase activity, where we actually measure the amount of enzyme that's present and its activity by coupling it to an exogenous electron donor in a chemical environment. And that's very important because it gives us a look at how much potential the algae could have to actually produce hydrogen, not just how well that potential is being coupled to metabolism in vivo system. So the reaction that it all comes down to is this 2 protons and 2 electrons to 1 hydrogen molecule. And surprisingly, there's really only 4 classes of enzymes in nature that are known to do this reaction. But many, many subclasses, of course, variations. So the nitrogenase is an enzyme which fixes dinitrogen out of the air into ammonia or nitrate and so forth for incorporation to amino acids and basically fixing nitrogen. It's very important, but a byproduct of its activity is some hydrogen production. That's usually found in bacteria and not found in green algae. Similarly, nickel iron hydrogenases are a type of hydrogenase enzyme which makes hydrogen but uses a different type of metal center, a nickel iron center, than iron hydrogenases in green algae. There's also a metal-free type of hydrogenase which has recently been called the iron hydrogenase, which is only found in archaea. So green algae only have these iron hydrogenases, and they're incredibly active biocatalysts. And what's interesting about them relative to other catalysts that a lot of people study at the School of Mines, like platinum and so forth, is that they're composed of readily available minerals or elements like hydrogen, carbon, nitrogen, oxygen, that are very easy to find. And it's amazing that biology has found a way to use these common elements to do this catalysis. So I know this looks really complicated, but I'm going to break it down into individual sections. This is basically a breakdown of where hydrogen is produced from in the algae. And for those that aren't familiar with this, what this is basically showing is potential here, so electric potential, and light coming into it. So everything along this is basically either proteins or protein complexes, which are in the chloroplast. And the little lightning bolts here represent light coming into the system. This is so-called Z-scheme microengy. And what it's showing is that light comes in, allows for the splitting of water and generation of oxygen, protons, and electrons. The electrons are then carried from protein to protein through this photosynthetic electron transport chain. And as they go to a lower energy level, actually there's a second photosystem, the twin engine of photosynthesis, photosystem 1, because it was the first discovered. And more light comes in, is harvested by chlorophyll, and the electrons are raised to an even higher energy level and passed on to ferredoxin, which is a soluble protein in the chloroplast, which can then transmit these electrons typically to an enzyme called Visco for carbon fixation. But in this case, during transition from anaerobiosis or anoxia to normal photosynthesis, it can temporarily shunt these electrons to the hydrogenase. And so basically couple the splitting of water to the production of hydrogen. Electrons can also come into the system through a second mechanism where instead of directly from splitting the water, there's an intermediate in this whole transport chain called plasticlinum. And it can actually receive electrons from the breakdown of starch. And during fermentation or the use of acetate by some algae, these electrons can also feed into this electron transport chain and be shunted to the hydrogenase. Lastly, the breakdown of starch itself through normal fermentation can also reduce ferredoxin, and in the dark reduce the hydrogenase and lead to a small amount. Although this reaction is much slower, and we only see about 1% of the instantaneous activity that we'll see during photoproduction. And of course, it's kind of a roundabout way of producing it from water, because you have to go through the whole process of making sugar in the first place, and then breaking it down and fermenting it. So to get into these specifically, so these are the light pathways, as I said, and so it comes right in. But then the dark pathways are much more complex, specifically Chlamydomonas reincardia, which is the first algae which is genome sequenced, and probably the one with the most research done. It has a very complex heterofermentation, unlike higher plants, which can only produce lactate, or ourselves, which have very complex fermentations. The algae actually can produce many, many different compounds, glycerol, lactate, ethanol, acetate, and they really focus in on where the hydrogen production is coming from. The breakdown of these starch and sugars goes to an intermediate called pyruvate, and that pyruvate is then decarboxylated in the reduction by pyruvate ferredoxin oxoreductase. and reduces ferredoxin, which then can donate these electrons to the hydrogenase and catalyze the production of hydrogen. And when I first came to Colorado, working with Paul King and Marie Berardi, we actually did Northern blots to show that P4 was expressed simultaneously to the hydrogenase A1 and hydrogenase A2 in plant hormones, whereas it was not present during normal aerobic growth. So this was the first indication, based on genomic evidence, that this pathway does occur in algae. This is the source of electrons for dark fermentated hydrogen production. This is important because this has to happen as a step to produce the next intermediate, acetyl-CoA, for the production of acetic acid and ethanol as their normal fermentation. The other the next thing is how do we actually measure these different pathways? And so this is important to understand some of the data that I'll be showing in a moment. And basically, we have specific measurement methods for in vivo and in vitro assays. So in vivo, we can actually measure photoproduction through this, which I have back in the back of the room, a Clark electrode. So in this case, there's actually two probes, and you can see the algae in here and the active cell. And we can basically measure this polarographically by reduction or oxidation of hydrogen. We can also use sensors, which I'll talk about in a moment, which turn blue in response to hydrogen for more high throughput screening. And dark fermentated hydrogen we can measure with a gas chromatograph, which I won't go into great detail, but it's very typical gas chromatography, which you take a headspace sample and inject it onto the column and measure it by thermal conductivity. The other way that we measure in vitro activity is by providing an electron donor out from outside the cell. So the cells are basically quasi-lysed in a Tripenex detergent solution, and electrons are donated by a dye called metabolism. And the amount of hydrogen produced in this assay can be measured by gas chromatography, or you can do the assay actually in the Clark electrode to measure rate of production. So I'll get into the specifics of the Clark electrode. The Clark electrode we use is this YSI 5331 electrode, and it has a platinum side and a silver side, and they're connected through a device which has to provide a polarization and measure the current as it travels through. The current is directly related to the chemical activity happening in both the anode and cathode on this electrode. So in the case of oxygen detection, you're having the oxidation of silver coupled to the reduction of oxygen, producing some water and silver acting on the other side. In the case of hydrogen detection, the polarization can actually be switched around. Now hydrogen is being reduced, hydrogen is being oxidized and silver is being reduced. And in that case, the flow of electrons is going the opposite way, essentially. We can still measure the current and then directly relate that current as a voltage to the amount of hydrogen or oxygen present in the solution. And this is very convenient because you can also get a rate from this production. So you can shine light onto the cells as they're in this chamber, and the electrode, as it measures the change in concentration of gas, can then be used to determine the rate of production. Unfortunately, everything on the market that does this would not work for our purposes. I'll talk about that in a minute as well. This is from a patent from someone in the room. And basically what this shows is another type of way that you can measure hydrogen photoproduction in a much higher throughput. So this basically is glass with a layer of tungsten trioxide and palladium, and then a layer of Teflon to protect the tungsten trioxide and palladium from water. So that you can actually take these glass plates and put them on top of algae colonies, shine light on them after they've been anaerobically adapted, and then the production of hydrogen from each individual colony will then turn the tungsten into blue reduced tungsten. So then you can visualize the production of hydrogen. This allows for high throughput screening for those that aren't producing hydrogen, which I'll talk about why that's useful. And lastly, I'd like to talk about the in vitro assay we typically do in the lab. So I mentioned a couple times that the iron-iron hydrogenase, while possible to be reduced in vivo by the photosynthetic electron transport chain or by fermentative activity, that we can provide an exogenous electron donor. So there's a commonly used dye called Metabolism, and when it's reduced, it actually turns from clear to blue. The little device that we have in the back here is one that I built with Ted Carnes, who's here today. And it uses actually an electrode system to reduce the metabiology directly by electrons to the dye. But more typically, a commonly used reducing agent, sodium dithionite, which is shown as the sodium salt here, is used to reduce in a basic solution, basically. Is allowed to reduce the metabiologen. And then the metabiologen, which has a reduction potential more negative than hydrogenase, can then reduce the hydrogenase subsequently and provide the electrons driving the production of hydrogen from protons in solution. So this is a convenient way to assay the amount of protein in solution, whether or not it's coupling well to biology. Okay, so what does this all mean? So I'm finally getting to what I did. This was something that basically informed us when I first got out here. And this was a study by Brandwright and Leanne. They looked at hundreds of algae and their hydrogen photoproduction capacities. And there's really just a few outliers that had high rates of hydrogen photoproduction, similar to Clammomonas reinhardtii. Similarly, there was some recent work by Thomas Hoppy's group in Germany, which also confirmed exemplary in vitro activities in Clammomonas meussii. So one of the first things I did to get a hands-on feel for these different assays and just kind of initial project was to survey some different strains, some different algae strains for their hydrogen photoproduction capacities, and to validate what others have done. And what we found is that, similar to Thomas Hoppy's work, Clammomonas meussii has very exemplary, under this temperature, which is 25 C, in vitro hydrogen production activities. So Clammomonas meussii, I learned a lot about in this research. It's interesting because while this strain here, 24.91, has exemplary activity, some of these activities were lower from other strains of Clammomonas meussii. These other strains came from the UTEX culture collection, whereas 2491 came from Europe. Although all of them have a morphological trait which makes them Clammomonas meussii. In addition, we looked at their 18S ribosomal RNA. But all of them, instead of having a single paranoid like you see here, this is a cluster of proteins which is associated with fixation of carbon dioxide. Meussii has two. And so not only do we have the evidence based on its ribosomal RNA, but also morphological evidence that we'd also use for things like higher plants to characterize it as Clammomonas meussii. And so we have 2491, UTEX 10, UTEX 96 are all different Clammomonas meussii that were reported by Brandon Wright and Hoppy as having higher activities. We also looked at UTEX 1060, which is a Clammomonas coleus, which also had reported to have higher hydrogen photoproduction activities back in 1989. Well, what we found interesting is that when you compare the in vitro hydrogen photoproduction activities to the in vivo hydrogen photoproduction, it's Clammomonas reinhardtii still performs the best, still have the highest activities, although these other meussii strains all seem to have an earlier induced hydrogen photoproduction phenotype. One thing that's interesting about this is that Clammomonas reinhardtii is the only of the four strains that can utilize acetate, which is in the media in all cases. We brew these all in the same media. The acetate not only contributes to the photosynthetic electron transport chain by the reduction of plastoquinone, there's also been one study saying that it potentially decreased photosystem 2 activity. So the fact that the different meussii strains were unable to use acetate may have an effect on their ability to actually do the in vivo photoproduction we're interested in as a source of fuel. However, we also looked at their ability to produce hydrogen under sulfur deprivation techniques. We still found that meussii did not perform as well as Clammomonas reinhardtii, even when we used a media that had no acetate at all. So there's something about the fact that although it has high activity when coupled to metabiologen, it's not coupling to the photosynthetic electron transport chain the same way. It's not wired in the same way as Clammomonas reinhardtii. So it basically showed some drastic differences. We also, this was when I first got into making sensors with vapor deposition in Poswitz's lab, we were able to show that the sensors themselves could be used to assay the active hydrogen photoproduction activities and do similar work to Brandwright and Leanne on a higher throughput way from some of these culture collections which house thousands of strains. The other thing, so we went on to further characterize hydrogen photoproduction in these strains by another method, working in collaboration with, for instance, Nadia and Anya, who's now directors in Chuckus mucis group. And this is a little different. In this case, we actually flash the algae for about a minute after they've been induced for the time specified here, and then looked at the hydrogen photoproduction as possible. So what's the rate of production for that short period of time. Under these conditions, we actually flash the algae with just enough light that they're doing some water splitting, creating reductant, but it's not so much that oxygen is building up in a solution, so they remain anoxic the whole time. Under these conditions, it's 10 seconds of light followed by 90 seconds of darkness. So the areas in black here are actually pulses going up and down, and so they're compressed together over the time period shown. That's why it looks like one big black area. A couple things of interest. What I found interesting is that C through F, which were all different strains of Chlamydomonas mellucii, had a strain-specific response to anoxia, and they induced their highest level activity early on, which I found very interesting, even though their instantaneous rates varied a lot from the previous type of assay. Also, Utex 1060, the Chlamydomonas coleus, which had previously been reported by Brandon Wright to have high hydrogen photoproduction activities, but in my conditions didn't seem to do much at all. It's the one basically flat line here. Under conditions where the photosynthetic electron transport chain is constantly provided with reductant, it seems to actually increase in activity over time, as if there's some sort of response or regulation mechanism that's going on. So I found that interesting as well, but I don't have another species to compare this to see if this is a species-specific trait. But in Chlamydomonas reinhardtii, we see a similar induction pattern as we would see under normal dark anaerobic induction of hydrogenase activity. It takes about 2 to 4 hours to reach maximum activities, whereas in the mellucii it takes only 30 minutes. So similarly, they had species-specific fermentation product production. Basically, Utex 1060 and the rest produced higher levels of ethanol early on and earlier induced activities, and we see species-specific patterns, but I won't get into this too much. But overall, we saw that these pathways were conserved in the green algae. So after looking first at this survey, around that same time we were finishing up that project, the second algal genome which had hydrogenase, so the Volvox genome had also come out around this time. But the Chlorella variabilis NC64A genome was released from the Joint Genome Institute. And this is an interesting algae. It's been studied for a long time because its ability to be infected by Chlorella virus. It also actually can be taken up by Paramecium bursaria and provide oxygen to the paramecium under zones where maybe it wouldn't normally have oxygen, so if there's light present. And also photosynthetic. So it's an endosymbiont of paramecium. But it can also live free living. We were able to get it to survive on a variation of the tap medium that we typically use for Chlamydomonas reinhardtii. The reason I was interested specifically in this algae is because when we looked at the genome, we found iron-iron hydrogenase. And at the time, there's really only algae in these groups which had been known to have hydrogenase activity, primarily by surveys of people like Brandon Wright and other people. And but in terms of molecular evolution, there was really only evidence of hydrogenases from chlorophycean strains. So at this point, there's 13 different hydrogenase sequences in GenBank. But it was the first, the Chlorella hydrogenase was the first from this class Streptophyceae, which is the next or ancestral class of algae. And what was most interesting about it is when we first looked into the genome, we found that it had these extra iron sulfur clusters. So these FS2, 4C, 4B, 4A are different iron sulfur clusters. And when you're looking at the sequences themselves, they have these motifs, cysteine, followed by another couple of amino acids, cysteine, and so forth. And these motifs are related to coordination of the iron sulfur clusters that are present in this internal domain. So it was the first algae ever to have these present. We ended up confirming the presence of these by sequencing the RNA, and which was tough. And we also did physiological work. In addition, we looked at phylogeny because it was interesting to us whether this hydrogenase, which is more related to very ancestral hydrogenases you normally see in Clostridia, Fermatoga, whether this was a product of a lateral gene transfer event or whether this was part of a nested hierarchy of evolution over time, which would be very interesting because if it was, that means that the loss was very late in algal evolution. For example, the embryophytes chloroplasts here, that's just a fancy word for land plants or vascular plants, are very removed from this very distal group that's been highly studied for hydrogen production. So if the loss happened right here, where I see loss of hydrogen clusters, that means that there's a lot of evolution that's happening where those were probably present. So, working with Eric Boyd at Montana State, we did a phylogenetic tree using hydrogenases, specifically just the H cluster of these different hydrogenases from lots of different species that contain iron hydrogenases, and determined that the chlorella was slightly more ancestral than these other known hydrogenases available, and intermediate between the diatom hydrogenase and the phyllosaurin. But together, these all grouped relative to bacterial or aphotosynthetic hydrogenases from eukaryotes that don't do photosynthesis. So this indicates that it truly is an intermediate form of evolution of hydrogenases and algae. We also found that these are reciprocally monophyletic, meaning that while hydrogenases are commonly found in algae, when we do find them, they seem to be coming in pairs. They're kind of 2 by 2. We see either 2 or 3 hydrogenases all together, as if there may be independent functions of each, which is one of the reasons we did the next project. So in terms of the physiology of this hydrogenase, one of the most important things we want to know is, does it couple to photosynthesis, and does it have greater oxygen tolerance? Because if it were more tolerant to oxygen, we could do higher levels of photosynthesis and produce potentially hydrogen simultaneously. And what we found is that in the pulse experiment, we see the same thing, hydrogen photoproduction, as well as we look at hydrogen photoproduction after periods of darkness, so after 30 minutes or 4 hours, 8 hours, or 20 hours. Although the rate was lower than we see in monosaccharide hardy on a chlorophyll basis. We still see fermentative dark hydrogen production. And of course, there's in vitro activity, but the in vitro activity is closer to what we see in monosaccharide hardy. So everything there checks out, basically. Hydrogenase seems to be active. It's coupled to metabolism similarly to monosaccharide hardy. But one thing that was very interesting is repeated work by Maria Gherardi looking at the oxygen sensitivity of the hydrogenase. When we followed her methods, we found basically that regardless of this extra N-terminal domain, the enzyme is equally as sensitive to oxygen as the truncated form of monosaccharide hardy. Similarly, we looked at the fermentative products and found that it had more or less a homolactic acid fermentation, although it did have the capacity to produce a lot of other products like ethanol, acetate, sesame. That it's mostly producing lactate, as shown in this modified figure where the large arrow here is in the lactate. But it has most of these enzymes annotated in the chlorella genome, GGI site. So it has these enzymes, and it seems to be that there is this nested hierarchy of evolution in this fermentative pathways in algae. What I got really fired up about, because these two are very close to each other, and it's interesting that it has an F cluster. But what I was really interested in, and have been since I was an undergrad at UC Davis, is these guys, the glaucophytes. They're the most ancestral algae known. They have a peptidoglycan wall around their chloroplast, and based on 18S ribosomal RNA, which this tree is based on here that I made, they're the most ancestral algae out there. And interestingly, I was really happy to be invited to look at some of the first genome data that hasn't been published yet. And when I took a look, it actually possesses many of the same enzymes that we see in Chlorella variabilis, Bolox, and Clang, as if it's been lost in other groups like Nostriococcus and higher plants, which have very limited fermentative capabilities. And further work will then look at phylogenetic trees like we've done with hydrogenase to see if these really truly are part of the nested hierarchy. They've presumably been evolving as long as the green algae have since the original endosymbiotic event nearly 1 billion years ago. So it's possible that some of these pathways came in from lateral gene transfer, as might be common in bacteria. But it's very interesting to me that this may be a very early event because it's the most ancestral algae known, which would point to the iron hydrogenase is coming in very early in polyphyletic tree of algae, and then subsequently radiating throughout some groups. So these primary conclusions from these diversity studies are that there's basically species-specific patterns in antioxidant production, both in time courses and the products they produce, and that there's in vitro activities cannot be assumed to correlate with these in vivo activities. So this provides reasoning to be looking specifically, doing screens at the in vivo activity. And that the F cluster was probably this N-terminal domain that's in these ancestral hydrogenases was probably lost very late in algae evolution, the divergence of Coryphyceae and Tragophyceae classes. And that these are, despite possessing that domain, it can still be linked to photosynthesis. So loss of the F domain is not requisite to be coupled to photosynthesis, although activities were a little bit lower if we further looked into why. And that the algal iron hydrogenases still appear monophyletic, so there's not a bunch of independent gene transfers that happen. A lot of other microbiologists will tell you that in bacteria that it's very common to have lateral gene transfer, so it's an open question as we get more genomes available as to whether typically algae do a lot of gene transfer or whether it's in more of a nested hierarchy as we see in higher plants with specific classes of engineering, specific metabolic capabilities. And then lastly, that these early heterofermentative capabilities, we have some new information that these likely date back to this early algal evolution, and there were subsequent losses in other groups like the metavascular ones. All right, so the other project I worked on specifically looked at Clamdymonas reinhardtii, which is the model organism that's been studied the most as an algal species. It has two paralogs, which is just a fancy word for copies of the iron hydrogenase. It has hydate 1 and hydate 2. It was already known prior to this work that hydate 1 seems to be the dominant hydrogenase in reinhardtii. But the role of hydate 2 is kind of unresolved. This is kind of interesting because based on sequence similarity, they're very, very similar. They have insertions, these extra insertions. They have all the required domains to be an active hydrogenase. But their level of expression is lower in hydate 2, and it doesn't seem to be coupled at as high of a rate. So a primary question that could be answered by studying Clamdymonas reinhardtii is what are the two roles of these individual hydrogenases? Is there metabolic differences and how they couple essentially? So are there specifically one coupled to fermentation and one to photosynthesis? And also give us some insights on what's the evolutionary pressure driving these gene duplications that we seem to see that may be happening independently in different groups of algae. So lastly, if we do knock out, or are able to knock out both hydrogenases in terms of mutating them, we provide a chassis for the expression of other hydrogenases, which may be more oxygen tolerant from organisms that haven't really been coupled. It makes a lot of sense that the Clamdymonas wouldn't have a hydrogenase that is insensitive to oxygen because under conditions where it would be doing oxygen photosynthesis, that would represent a huge loss of energy to just be doing amounts of nitrogen. But in our case, coupling an enzyme that's tolerant to oxygen would be optimum, would be ideal. So basically, the way that we did this knockout screen is we used the chemochromic sensors, which I mentioned earlier, and produced about 20,000 mutants in a background which already had hydate 2 knocked out from the growth hormone. This sensor work later on was used to develop an art project that went to the Museum of Modern Art. This is basically the ideal system to provide ability to screen lots of different strains, potentially in classrooms, for basically of the many, many different strains that are available. And we got a lot of pricing on these. But the reason it was possible is because of the development in the Posit's lab, where not only did we optimize the production of the sensor, but we also found that instead of Teflon, we found that we could actually spray on an alternative coating, basically from the hardware store. So polyurethane provided a much more stable coating, which lasted for a longer amount of time, which then we could apply to these glass stoppers, so that you could do various schedules of light and dark and potentially screen thousands of strains. So then, after we generated all these mutants, we did the physiological testing of the 30 that we found from the 20,000, which were unable to make hydrogen from the sensor screen. And then back-crossed those mutants, which had the altered phenotypes, and did gene walks to determine where the lesions were, specifically using PCR to look for which kind of. We have a lot of other interesting mutants that I won't talk about, but we were able to isolate 2 mutants in hydate 1 and map those insertion sites with work with collaboration with Sarah and then put the genes back in and show that we've restored function. And then finally, do the hydrogen physiology on all of these different mutants. So we found two different HIDA1s. One of them actually had mutations in the intron, in the 5' UTR. So it still maintained about 30% activity, presumably because the entire functional hydrogenase is still expressed at some level. The one that was completely knocked out in the intron here, still see a very small amount of activity. So because it's completely in the intron, it's possible that there's still some active transfer. But in general, it would work for our purposes. The way that these mutations were made is antibiotic resistance for paramomycin was inserted at the Grossman lab and knocked out HIDA2 using PCR-based screening techniques. And then we inserted zeaxanthin resistance, which confers resistance to another antibiotic, which is how we generate colonies for such things. So finally to the data that we generated. The wild type here, this is the activity of photoproduction. So after basically shining with saturated light after the algae has been anaerobically used for 4 hours, we get about 100 to 120 nanomoles per microgram per hour. And when the HIDA2 is not present, so HIDA1 mutant, this gets reduced down to about 25% activity, 20 to 25%. So this is the first time we show that HIDA2 is linked to photosynthesis, but at a much lower level. HIDA1, as would be expected, is also linked to photosynthesis, and can pretty much do most of the work by itself. And one thing that's still an open into question is whether this is being upregulated in this mutant in response to the loss of HIDA2. So, but for now, we're assuming that this is basically the breakdown during hydrogen photoproduction in the wild type. The two of them together comprise the total activity in the wild type. Under these conditions, we see no activity from photoproduction from the LP. However, we do see a little bit of activity by the MV assay, which is overrun with reductant. There's plenty of electrons available. We still see a small amount of activity. There's still an open-ended question as to whether another enzyme like pyruvate paradox-NOx or reductase may have some sort of hydrogenase activity in these conditions. So it hasn't been attributed directly yet. We have to do some protein isolation to get down to that. But generally, we see the same pattern, indicating that the level of hydrogenase present is reflected in the photoproduction activity that we see. And this also matches up well with previously work done on the transcript level by Meuser. When we look at dark fermentative activity, we see a different picture. We basically see that the HIDA2 is able to do the same amount of fermentative activity as the wild type or HIDA1. So there's such a slow or low amount of flux through dark fermentation that even when there's a very little amount of activity there, HIDA2 by itself is able to do most of the basic dark fermentative hydrogen production. So this may say something about evolution we may learn about more. And the W, we actually see a pretty good amount relative to the rest because, again, this activity is so low. Even the smallest amount of hydrogenase activity can be reflected in such a slow process. The most interesting thing from this work, I think, came from the complementation. We restore more of the wild-type activity. So what we have here is we have in vitro hydrogen activity, maximum hydrogen photoproduction, and the dark anaerobic hydrogen production. So we basically see that the in vitro activity is more than restored in this. This is the product, by the way, of two independent progeny that have been transformed with the hydrogenase gene that are averaged together in quadruplicates. So we've got 4 individuals and two progeny, and this is all averaged together, which would help us to resolve that this isn't some kind of strain-specific difference or individual progeny difference. And when we put hydrogenase back in, we see restored activity, or even more than restored, heightened activity. Interestingly, hydrogen photoproduction is not higher in the same backgrounds. So that seems to point to the possibility that the amount of hydrogenase that's in the native algae is already adapted for the electron flow coming from photosynthesis. There's enough there to handle the maximum electron flow after being flashed with light after the period of darkness. And then even if you provide more hydrogenase or more hydrogenase activity, you don't actually get higher levels of photoproduction. In the contrary case, we put in HIDA2, and even we looked at many, many different transformants, and we never found transformants that had restored activity of the wild type. So future work that would be interesting is to overexpress the HIDA2 or swap the promoters from the two enzymes to see if this is just a promoter-specific thing or if it's actually the enzymatic activity. There really is, despite high levels of sequence homology, something about HIDA2 that doesn't allow coupling of high rates of activity. Give us insights into whether high day 1 has something specific about it. Or do piecewise pieces of the protein make synthetic constructs which contain hybrids of the two enzymes to try to isolate what exact portion of the sequence is causing the slower activity. But a first initial thing next to do, I think, is to express these at a high level and look at the transcript levels of these two patterns. Of course, in the double mutant, it's just a negative control for this. So the primary conclusions is that we verified that high day 1 appears to be the dominant hydrogenase ingredient in C. reinhardti, and that high day 2 is similarly coupled, which wasn't as exciting as some other results, like that they have independent roles, like one's connected to photosynthesis and one's connected to fermentation. They can do the same activities, but high day 2 does it for some reason at a lower level. We also found ultimately that the algal hydrogenases are monophyletic with indications of multiple independent gene duplications, and that given these similar activities, there's really no indications yet as to what the selective pressure is driving this high day gene duplication. Although it seems that there's a concentration of high photoproduction activities in chlorophyceate, it's a very distal group in algae, and that's where people have focused most of the molecular work as well. And that there still may be some kind of correlation between loss of the duct cluster and these high photoproduction rates that we see, although we haven't resolved exactly what it is. So work in progress is we'll express oxygen tolerant hydrogenases in this background, look at hydrogen localization, and I think it would also be interesting to continue to do diversity work, looking at the genomes and comparative genomics will provide some interesting details if you combine it with physiology. So I'll quickly go through the last thing, which is this Clark electrode system that we developed to do these measurements. These measurements and some of the other work that I've done were all done with the Clark electrode system, which could not have been done without a lot of work by Devin Carnes and Ed Dempsey, and I'd just like to acknowledge their support on that because they're sitting right here. And because really I could not have done this alone. They basically together did the software and a lot of the hardware on the system. And what it is basically is a revised, a more advanced system to measure hydrogen production that keeps a lot of things constant, which are not kept constant in other commercially available systems, and provides now not available glass cells. So this kind of cell was originally made by Gilson Scientific. It's no longer commercially viable or available. And it allows for the insertion of 2 probes from either side seated with O-rings. So you have a completely sealed environment. A lot of the systems that exist that are made out of plastic right now are not impermeable to oxygen, so they're sufficient for respirometry data or photosynthesis measurements, but for hydrogen production measurements specifically, we had to work with ASG up in Boulder to get this similar cell built. And it has an airlock basically to keep it completely isolated from the external oxygenic environment. A lot of other things went into this, and that is specifically in lighting control, because this is an LED system. As LEDs heat up, they actually decrease in photonic flux at output. And so working with Ed and Devin, we actually put a Peltier cooler on the backside of the LED to either heat or cool the LED to keep it constant. There's also a receiver that receives the light and uses the circuit to actually control the output so that it's controlled. This way the user can, in software, calibrate the light across make a calibration, and then set a scheme for the intensity of light that they want, and for the amount of time that they want that intensity, and they can know that it's going to be that intensity. This is going to be very difficult with an incandescent system. We also have temperature control from 15 to 45 degrees C, similar to people who have done PCR, a thermal cycler as a Peltier cooler on it. And in software, we've also developed corrections for salinity and altitude. On the actual circuit board, there's a circuit which measures the pressure at whatever altitude you're at. And then back in software, we calculate based on calibration gases that you're using, the amount that's there. And we have developed user-calibrated analysis variables for units that are typically used for algae work, like the number of cells, chlorophyll, volume used. The calibration can be done based on PAR, so you actually graph the output, you can do lumens or whatever you like, at whatever spectrum you like. And then you can set up a user-defined experiment, so dark, light, flashing, whatever you like to do. One of the most interesting things about this, though, or helpful things compared to other systems that exist, is people typically get a system designed for measuring oxygen and then adapt it for hydrogen measurement. Those systems don't have the amplification of the signal that you need to measure hydrogen. But hydrogen signals from these types of systems are much lower, about 100 times lower. So typically what's done is that polarization is actually alternated to make this sensor more sensitive by putting on what they call platinum black, which is a little bit of voodoo in terms of what exactly that means. Our system doesn't require any of that polarization alternation. You can start measuring hydrogen instantly because it has amplification using advanced electronics that are available now. And we've validated, this is from Devin's thesis, we validated that it provides a linear response up to 10% hydrogen gas. And it's very stable because when you do the polarization, alternation of polarization, it makes it very sensitive. But as soon as you start doing measurements, you lose sensitivity very quickly. So your calibration is constantly changing as you're trying to do a time course. In our case, because you don't have that platinum black, you don't lose the sensitivity as quickly, and you have a much more stable system over time, which makes it much easier to get reliable data and lower errors. And this is an example of what you'd see in a typical experiment. This is the rate down below, which is being calculated based on the chlorophyll that's being added, the sample volume, calibration gas to use, the altitude. In this case, the altitude is typed in. In the model you see back there, it's actually, again, has that circuit where it actually measures the partial pressure. So if a storm comes by, like the storm we had a couple days ago, it would actually compensate for that in real time and know exactly how much gas is present in the solution, depending on the actual pressure. So this is basically a period of darkness. The light comes on, you see a burst of hydrogen being produced, the light goes off, and then you see the uptake of hydrogen, which also occurs in green. You can do a 2-point calibration by dragging bars across wherever you like, and you actually get a rate determination based on these variables that you input. You can also export the data in Excel form. That's it. So I just want to, I have a huge number of people to acknowledge. I've really been the ear of a lot of folks over the years, and a lot of people have been nice enough to help me, both in the lab and in other ways, from a lot of different places. So I have a huge amount of gratitude and thanks. I'd like to particularly thank Matt Poswitz for his enduring support of ups and downs, and continual funding is always helpful. John Spear has also always been there. I kind of barnacled onto John's lab before it was Diane Almond's lab. He came in, I didn't want to leave, and John was nice enough to let me stay and keep working, despite the fact that I work on things that are very different, somewhat similar and very different. Similarly, Maria and Mike have been there for me throughout this process and been extremely helpful in providing openly their amazing knowledge in this field. And I'd like to thank the rest of my community as well, and all the other people in the lab that helped throughout this process. And got a lot of funding, and that's it. So. Questions first from non-committee members, and I think the committee wants to go.
Thank you very much, John. I just want to add a few things before we do the Q&A. As you mentioned, it was certainly a very interesting path through here. But before it was all finished, Jonathan has co-authored 8 manuscripts. There's 2 more in review. We have 4 more at various stages of write-up. And he's got a suite of mutants which are in various stages of development. He was also part of 2 patent applications that have gone in and just turned from what I think is a successful synthetic genomics continuing studies in San Diego. So it's been a real pleasure having you through, John. It's never been dull. lots of good information. A lot of the research also that you put together for our laboratory has gotten us another 3-year grant into the final round of another grant. So it's all very good. It's been very, very nice to have you. I think that we can take some non-thesis committee Q&A and go from there. Okay.
Good job, John.
Thanks.
So I have a couple of questions. The first one pertains to the first part of your talk with hydrogen metabolism. You banter back and forth using the terms metabolism and catabolism for the production of hydrogen, and it's an energy-intensive process, correct? It takes energy to produce hydrogen.
About 420 moles.
So that's an anabolic process, right?
I don't believe I said catabolic. Maybe
You used that term a couple of times.
Catabolic sugar.
Okay, so that's probably just a reflection of mine.
So they're linked together. So during fermentation,
Right.
breaking of the carbon bonds has to be linked, has to be coupled to electron acceptors. So in the case of the decarboxylation of pyruvate, hydrogenase is acting as an electron acceptor and then coupling that into proton production.
So then the benefit to the organism then is, why does it need to produce hydrogen, I guess is my...
Well, the main benefit is that just typically through glycolysis, it has to produce NADH and ATP. And so that you can basically cycle, normal processes, produce the two energy currencies of the cell. And to do that, throughout the process, you have to break carbon bonds along the way. And so it's an intermediate step in producing production of acetyl-CoA. You can also do it through another mechanism through PFL, which produces formane. But it's a commonly used anaerobic shunt, to get to acetyl-CoA. So then you can get to ATP generating steps where you produce ethanol or acetate. So it's an intermediate step leading to another step, which is going to produce phosphate bond, which can be used to run your anabolic process like you're seeing. So you have to do the catabolic process and then couple to typical anabolic growing. Maybe under fermentative conditions, which is tougher conditions.
So my second question relates to the second part of your talk where you were talking about the genetic diversity of these unicellular organisms. And you made a statement comparing unicellular algal diversity to multicellular or vascular plant diversity. And that kind of popped a question into my head. You were talking about whether, the uncertainty of whether there's any kind of gene transfer that's going on